So before I start, two things. So one is I would encourage people to stretch or stand up because I don't think you get more breaks. So I don't want people falling asleep on me. The other is, can we get someone to dim the lights? I've got a lot of fluorescent pictures that are not great with backlighting if possible. I know Craig did it before, so it is possible. You don't want to stretch? No?
The right one. The left pedal, the right one.
There we go. So here we go. Doesn't that look better in darkness? I look better in darkness too. So I'm Aaron Schindler. I'm visiting from Sydney, so it's a bit of a commute for me to get here. And I will preface this by saying I'm certainly not an XLH expert, but I am a gene therapy person. And certainly for the conditions that I work on a lot, which is brittle bone disease and NF, there's, from the patient communities, there's often a lot of inquiries about where are we up to in terms of gene therapy? How long will it be till a gene therapy cure? And I tend to be very positive about the potential of gene therapy, but I also try to balance my responses by the idea that this is a way away, 5 years, 10 years away before actually seeing things going into clinical practice. So I think it's important to balance the hope with some realism about where things are going. So I don't have any disclosures. So my talk today, and I've been told that I have to sort of speed things up a bit, but I'm going to talk a bit about sort of where gene therapy is. Then I'm going to talk a little bit about AAV vectors, then a bit about CRISPR, and then a bit about what we're specifically doing in the gene therapy space and how XLH might fit into that. So gene therapy is the idea of where, rather than trying to treat the symptoms of the disease, we're going right to the genetic mutation itself. For people who are aware of gene therapy, it has a bit of a checkered history. So going back to some of the SCID trials from the early 2000s, there are cases of cancer that have been related to gene therapy that still cause concerns today. Gene replacement, though, recently has had some quite big successes with things like Zolgensma and Luxturna being quite transformative in terms of what they've been able to achieve. But although there's a huge amount of talk about CRISPR, and when you listen to the news, it seems like everyone's going to be treated with CRISPR and fix everything in a year or so. But there are no CRISPR therapies currently to market. There are a huge amount of trials, but there is a distance to travel before that occurs. But I think people need to come to terms with the reality that there is a massive amount of investment going into this technology, and I'm certainly of the belief that it will go somewhere, in those sort of 5, 10, 15-year time periods. So before I start getting into what we're going to talk about, I just want to differentiate between the idea of gene replacement, which is really a form of gene addition, and gene repair. So with gene replacement, what we're talking about is expressing a deficient or absent allele in key cell types. So that can be very, very powerful when something is missing. But for diseases where you have a dominant negative, and admittedly this isn't necessarily the case for XLH, but when you have a poison protein, and we do see this in brittle bone disease, you have a limited ability to treat something by adding something in. It can be very difficult if you're using things like AAVs, if you have gene overexpression, with large gene sizes. So the vectors we use have packaging sizes. If your gene overexpression is a problem, that's also bad. And we're always concerned about the idea of insertional effects, both of the gene and just generally of viral vectors. So CRISPR allows you to potentially get over some of those hurdles of gene addition by repairing the DNA, and many of the sort of newer CRISPR strategies don't even involve double-stranded breaks. So you're minimizing risk to DNA damage. So we can restore ideally native alleles, but there are similar challenges that exist for delivery, particularly for things like bone. And we need to look at issues of efficiency. So some of the approaches to being able to edit things with CRISPR are very powerful, but at the moment very inefficient and probably not the levels where they're going to be clinically translational. So our vector of choice that we tend to work with are AAVs. They're generally considered fairly safe, and I can talk with people afterwards more about things like liver toxicity, but they're generally fairly well tolerated. They're very versatile, and they can be very efficient. So, I mean, there are other options, but for things like bone, probably AAVs are going to be the way that we go initially. So there are a range of different serotype types, and the way that we target AAVs is by the capsid protein on the outside. So the capsids that we play with are specific to different cell types. And then we usually provide some sort of promoter sequence if we want it to add extra tissue specificity. So I guess I'll sort of talk about how we've been doing this, and we use a reporter mouse model called the AI9 mouse, which is a conditional TD tomato expressing mouse, so it's a fluorescent protein. I, as a scientist, for some reason, really like results I can see with my own eyes, which is why I like fluorescence. So in this, the mouse is not fluorescent until it sees this Cree protein, and then it excises a stop cassette, and then you get fluorescence. So we went through and decided to test a whole bunch of different capsids, and we have a paper published where we did this, and we did this by injecting locally into fractures a whole bunch of 20 plus capsids, and found AAV8 was particularly good. So then we took that AAV, which, sorry, this is sort of what we do. This was in the fracture, and you can see sort of how we inject things in there. For the matter of time, so we took this AAV vector that we know goes well into bone, and then we applied a bunch of different promoters. So you can see here, do I have this pointer work? Okay, so we have the CAG promoter, which is a constitutive promoter. We have these Col 2.3 and SP7 promoters, which are different, theoretically bone-specific promoters. So you can see here, this is the non-specific promoter. There's a bunch of staining here in the muscle, which obviously, because we have no bone specificity, which seems to go away when we start limiting to bone. But you can see all throughout the bone, all these red cells. And this is, by the way, a single injection of the AAV, just once. And it was not considered a high dose by any means for what we do in a mouse. We can do 5-10 times higher. So then we went and looked at other tissues to see whether that was actually specific. And you can see with the CAG promoter, a huge amount of expression in the liver, and that's not unusual. AAVs do like the liver. There's some in the heart as well, but all throughout the other organs. And we got increasing specificity. And here with the SP7, this was our sort of best one, where we looked at all these other tissues, and we didn't see it elsewhere. I did get a question before in a break. Is it in the odontoblast? I don't know. Odontoblasts are SP7 expressing cells, but we didn't specifically look. So this is a sort of newer result, which is unpublished, looking longitudinally, and that's sort of the paper, what's going to go in the paper, but I just zoomed in on these photos just to make it a bit easier to see. So what you can hopefully see is that when we start 1 week after we've transduced these mice with the vector, most of the staining's on the bone surface, but then over time what you see is the osteocytes inside the bone start becoming staining. So what ends up happening is we're getting all of the progenitor cells on the bone surface being affected by the therapy, or the transduction. It's not really a therapy, it's with the vector, but then the population of osteocytes inside the bone are gradually populated. We are looking at, as I will talk about later time points. So one of the questions is, you see a huge number of cells there lining up in the bone. Is that enough to actually have an effect? And that was one of the questions that we had. So we decided to use a system that we had available to us, which is this SOST FloxFlox mouse. So SOST, sclerostin, is a key negative regulator of bone growth. When the gene is missing, it's associated with a high sort of bone mass disease, sclerostosis, also von Buchan's disease. And from people knocking out the gene, there are quite big, profound developmental effects, which are also very difficult to tease out when you're looking at postnatally. So we thought a system to try to create the inducible knockout. So we used our AAV8 SP7 pre-vector in those mice. So what you can see hopefully here is both brighter and wider bone staining. And what we did is, this is even after sort of 2 months, where basically we're starting to get good osteocyte expression at this stage. Even by this stage, we're getting quite significant increases in MAR. So what this is showing is that the amount of cells that we're able to affect with these AAV vectors are able at a genetic level to have profound effects on bone. So we are hitting enough cells to do something. So sort of to wrap up this work on the AAV vector, it shows high efficiency and specificity for bone. It persists for at least 8 weeks, and we're doing longer studies to address that all the way out to a year, year and a half. Where it affects progenitor cells, which become osteoblasts and then osteocytes. And just people who want to use it in context of other flux mice they have, you can use it to create bone-targeted knockout mice using those strains. And for people who want it, we are giving it away. It's not a secret reagent. So we're happy to collaborate or supply that. So with the SP7 promoter, I guess the question that we are thinking about moving forward is bone specificity needed for repair scenarios? So sometimes if you're trying to repair, if you're doing a repair event, you don't necessarily need to just hit the bone. You can repair it in other tissues, even if it's not expressed there a lot. Could this technology also be used to target other tissues? Yes, I love bone, but I also like other musculoskeletal tissues, and we've been working on, so this is a muscle-specific one. Interestingly enough, the sort of vectors that people are using for current muscle targeting for gene therapy for things like Duchenne's, most people will look at those genes and then they'll look at the muscle and they never look at bone because bone's really hard to section. But we look at some of those and they don't show any specificity for muscle. Well, they're in bone as well. So we're trying to create some that are truly muscle specific. So can we integrate this with CRISPR-Cas9 editing? So we've been playing around with, I say playing around, it's been a big journey to get the technology working reliably for us. CRISPR for about 5 years. And this started as part of a work done with the endocrinologist in the hospital, looking at a bone fragility cohort. So these aren't brittle bone disease kids. They're often children and teenagers who have above average numbers of fractures. And so they get sequenced for gene variants. And a lot of these variants are classed as variants of unknown significance. So what we've been doing is trying to create models of these or cell models of these by introducing the mutations or disrupting the genes and cells. And here you can see some of the graphs that we have after we've applied CRISPR to human cells to try to introduce these things. And what you can see is disruption is a pretty efficient process. If we try to do HDR, which is homology-directed recombination, to try to introduce specific mutations, that is a lot less efficient. But one of the good things we do get is that the outcome measures we get is we get pretty much every sort of mutation and what sort of prevalence it has in our cultured cells. And then, something which is 14%, we can easily pick clones and then grow up those clones to get clonal cell lines with the mutations of interest. So gene disruption in cell lines is straightforward. HDR is more challenging. I think those are the take-home messages here. So what are we trying to work on with our AAVs and CRISPR? So one of our big goals is to try to find treatments for osteogenesis imperfecta. And one of the ones we've been working at the moment is a severe OI mutation. It's a 20 base pair deletion. The child that it's associated with had about 19 fractures in the first year of life. So quite a severe case of OI. So what we've been doing is creating models of this deletion in mice to be able to test therapies and test their therapeutic efficiency. Because efficiency, hitting the right level of efficiency is going to be key here. So we've been creating the human cell lines with the delta 20 deletion. I guess subsequent to me sending these slides, because I had to send them early, my lab told me that they have got the delta 20 deletion, so we've succeeded at that. So the next step is to now use approaches to try to deal with that. And we really have two. The first is to use what's called a nickase approach. So instead of cutting both places, you cut in two places, and we'll cut one around where the 20 base pair deletion is, so we'll only hit that allele, and one upstream to delete a large chunk of the gene. And that will turn, hopefully, a poison protein negative allele into a mild case of OI. The second is to use what's called a prime editing approach, which is using a reverse transcriptase stuck to the Cas9, which allows small regions to be edited. So we've also created a mouse line with the same 20 base pair deletion in its call1A1, which is currently being bred up. And the idea is that we can then apply the vectors to that mouse and see what efficiency we can get and see what phenotypic reversal we can get. So This is actually quite an efficient process in terms of both creating mice and cells, so it's in the scheme of things not high cost. So I guess I sort of wanted to put a slide in here about my opinion of where I think gene therapy could go for XLH. So the FEX gene is big, and when you have big genes, it makes that in the harder basket for gene therapy. So in terms of approaches, gene replacement is hard because it is big. The approach, which obviously we've heard about today, of expressing inhibitors, is a clever way, I think, to get around that issue. But it may be difficult to ensure that balance of FGF23 activity and regulate that over time. And I think all of these sorts of replacement therapies have to also be taken in the context of the fact that burosumab exists. And so, do you want to risk a therapy when there is other treatments that can be made? I think longer term, we will be able to work on gene repair strategies of actually targeting the FEX gene using approaches like AAVs that target bone. I think what you're going to see is people, again, targeting the low-hanging fruit. So single base change mutations with base editing enzymes. So there are base editable Cas9 variants that don't cut the DNA. They just change single bases, and they're quite efficient. And so that sort of thing I think you'll see first. Oh, wrong button. So just sort of end in terms of what we're trying to do and where our collaborators are with this is that, as I said, AAV vectors are limited with their packaging size, and that can be a real issue for CRISPR. Some of the bigger, newer enzymes don't fit very well. When you try to split things across multiple vectors, again, people can talk to me about this afterwards, there are a lot of issues with that. So what we've been trying to do is work with Samantha Jin and Gaten Bergio to create new tools to do this. So basically creating smaller versions of these enzymes to be able to fit them better into vectors so we can do things with single vectors. And I think that's a really exciting area where we can add a lot. So I'd like to thank all of our funders, particularly the TQ Foundation, who has been quite generous with trying to support gene therapy for OI. And often I think basic research can be challenging to try to fund, and we really are grateful to our philanthropic donors. I also want to acknowledge the people who do most of the actual lab work, particularly Lucinda Lee and Alex O'Donoghue, who did, I think some tremendous work in both the CRISPR and the AAV work. I think I managed to get that done in about 20 minutes. Am I okay? A thumbs up. So maybe I'm allowed 5 minutes for questions, because I'm sure there might be a few.
So I missed exactly, was it just a pure attempt to find an AAV that was bone specific, or did you modify the capsule in any way to make it bone specific?
So we didn't modify the capsid. We ended up screening a lot of capsids to get to that one. There are a few. That was one that we thought was the best. It did work in human cells as well, but I think for humans, the AAV DJ seemed to work the best in human cells, but didn't work quite as well in mice.
So there's no way to, put on a long arginine stretch to sort of drive it to bone or anything like that?
They don't tend to work like that with the capsids. So people do a lot of, we work on NF as well, which affects the Schwann cells. And our collaborator, Samantha Jin, has been working on using what are called capsid shuffled libraries, where you mix them all up and then try to select away good capsids for other tissues. So the picture that I had up there for muscle, where that used AAV9, that's the conventional one. Like the day before I left, again after the slides, my student sent me AAV Maya, which is sort of the new one that's just been published for muscle, and it's completely bright red, blowing my eyes out because it's the new capsid for that, and it works very, very well.
Thank you very much.
Yeah.
Is there any way to get the gene as well, or the gene repair as well, to the teeth?
So the answer is I haven't looked at the odontoblast yet. I think it's harder to do that histology. Yes, enamel in the way. So I was talking to the earlier speaker about potentially collaborating to get someone to help, because we can send samples over to get them sectioned. So the sections that we use are cryosections, but we use this Cornell method, which uses this special tape because it's got the mineralized bone on there to be able to hold the section from falling apart, but also not destroying the fluorescence.
It would be very important for later on, because if we have to repair bone only, then
Yeah.
Last one. Last question, sorry. So when you use gene therapy to target the bone, can you do that on children, on growing bones? Or because when you do gene therapy on the liver, then you need to have a big liver, so like just after 10 years.
So the answer is that we haven't done it in people. We do it in quite young mice. So the answer is I don't think that it really. I think you would want to do it earlier, but you probably want to base it around when you think it's important to be doing clinical intervention. So if the decision is that after the first year of life there's no difference in growth, you might not start till 1. So things like Zolgensma, which is used for severe spinal muscular atrophy, like that's used quite early because it's needed quite early. So I think that there's going to have to be some clinical decisions. The other thing is that we've been talking, at least in the OI space, is that when you get to doing these sort of things as gene therapy trials, it's really important to know what you want your outcome measures to be and have well-defined populations and patients so that you can do these trials properly. So we're doing the hard work as basic scientists, but as soon as this extends on, starts going in, starts looking towards patients, that's when all of the clinicians need to be, talking to us about how these trials are going to run.
I do not agree completely, because I think you can adjust your strategy. You can combine strategy. You could have a strategy for children and then have another therapy when they grow up. For example, the therapy for that goes through the liver is only possible when you have adolescents or adults. So you can use it only for adults and then have something. You can combine
I don't think there's any reason why this couldn't be used in adults. I'm just sorry, more answering the question of how young can you go. I mean, I'm from a kids hospital, so we tend to think a lot more about kids. But the question of how young you would go is often a big one.